Hi, hello, vanakkam, vandaram, namaste, namaskaram. This is your friend Wikidite and you're watching Wikidite YouTube channel. So, in this video, I'm just going to do an experiment. So, I'm just going to teach you how to extract a bacterial DNA within 10 minutes. So, most of you may use the conventional way of DNA isolation or the kit based way of DNA isolation for isolating the DNA, where the conventional way of DNA isolation will take minimum 1 day or 2 day. The kit based methodology will take nearly 1 hour or 2 hours. But in this video, I'm going to explain the DNA isolation procedure which could be able to be done within 10 minutes. So for doing this 10 minutes short DNA isolation, you need PCR tubes. Yes, of course, for the DNA isolation you need PCR tubes and PCR tube caps, and then this is a PCR tube caps and PCR holder. The holder with the tubes and everything, and you need a 200 ml tip autoclave and 200 ml or 100 ml pipettes, and you need your bacterial plates, the 24 hours grown bacterial plates, and exactly this buffer. This buffer is called as pig NaOH buffer. So I'll be giving the composition and preparation protocol in a description. You can find it, as well as I'm giving the Google Drive link where you can able to find the composition of this buffer as well as the preparation protocol. So you need this buffer and the 24 hours grown bacterial plates to isolate the DNA. So we'll start. You need autoclaved all the things before using this one. I'm taking 100 ml of buffer. I'm going to isolate DNA of 16 bacteria. So I'm taking up 16 PCR tubes with PCR caps, tube caps, and a pig NaOH buffer, and 16 bacterial cultures that have been grown for 24 hours. So if you're going to isolate the DNA, 1 day before you have to culture the bacteria in a plates. So here we'll be using the toothpick which is already autoclaved prior to the usage. So these are my plates. It's packed with the thin film, and so I have my grown plates here. So I'll be taking up a toothpick which is already autoclaved. So I'll be picking a single colony from this plate. I'll be picking single colony from this one. So I have taken a single colony, and in this one I'm just going to put in the first one. So I have made in the first one, and this place is done. Then the second one, where I'm picking the another toothpick and just trying to pick up a single colony for bacteria. Second one. Third plate. Third well. So I'm picking up this one. So fourth plate, fourth one. The fifth one. Sixth one. Seven. Eighth one. Close down, because I have taken a PCR tube with only 8. So I'll be numbering them. 1, 2, 3, 4, 5, 6, 7, 8. Other 8. The second row, first one. second row, second column. Second row, third column. சத்தியமாக பண்ண நினச்சது யூடியூப் ஷார்ட்ஸு, ஆனால் போகிறது 10 minutes வீடியோவாக போயிட்டுருக்கு. செகண்ட் ரோ, ஃபோர்த். Second row, fifth. Second row, 6. Second row, 7. Second row, 8. Actually, I have 17 bacteria. So now it's time to close cover. So I'm starting from 9, 10, 11, 12, 13, 14, 15, 16. As I have told you, I have the one more column for 17th culture. So here it is. So 17th is also over. So for the DNA isolation, what we have to do is, we have already prepared the buffer with the peg and the other composition. I'll be writing the composition and description, and we are taking the 100 microliter of that buffer and we are placing them into the PCR tubes, which is having nearly a 250 microliter of the volume capacity. So we are putting the 100 microliter of the buffer into the PCR tubes, and we are just picking up a single spot or single colony from the bacterial culture that you have grown for 24 hours and you are placing into the buffer. Now we have a PCR machine where I'm going to use the PCR machine for the isolation of the DNA. So this is the bacterial cultures that I have prepared for my DNA isolation. So I'm just going to place these bacterial colonies, which is already placed into the buffer, and I'm just directly placing onto them in a PCR machine. Now I have placed my 17 samples into the PCR machine, so I'm just closing the door. Now we have a predefined program for the DNA isolation where it is just a single step process, where you need to set up your PCR for the temperature of 80 degrees Celsius for 10 minutes. So you are going to place your bacterial colonies sample with the buffer in PCR machine for 10 minutes in a 80 degrees Celsius. So that's it. You can run the program. The PCR machine will run for 10 minutes at 80 degrees Celsius. Thereafter, you can use that buffer as a DNA template. Okay. So I'm just going to start it. So the sample that I have added is 100 ml. So I'm pressing 100 ml. That's it. So after the 10 minutes, you can pick up your sample from the PCR machine. You can use that sample as a DNA template directly for your PCR amplicon processor. In order to go for the DNA sequencing or in order to do any kind of molecular marker studies, you need a DNA template. So that DNA template, you can use this as that DNA template. We have added the bacterial colonies into the buffer. That buffer is already kept in a PCR machine for 10 minutes in 80 degrees Celsius. Thereafter, you can use that sample as a DNA template. So the buffer what we have added will lyse the cell wall of the bacteria and it will bring out the DNA from it, and you can use the DNA for the sequencing protocol or for any kind of molecular technique studies. So it is just a 10 minutes simple DNA isolation protocol you can do in your laboratory. And if you find the result suitable, just post your comments in the comment section. Thanks a lot.